Hello. Every day, millions of families trust cough syrup for quick relief. But after recent tragic incidents, one question now worries everyone. Could this bottle be contaminated with dangerous impurities like DEG or EG? But what is this DEG and EG? These are toxic substances used as industrial solvents and antifreeze agents. These substances can enter your cough syrup formulations when low quality or untested glycerin, propylene glycol, or sorbitol is used. The harmful effects of DEG and EG are severe and toxic, even in small quantities. Regulators across the world have set strict limits, not more than 0.1% in raw materials and finished pharmaceutical products, to ensure patient safety. But recent global incidents have shown how contamination can go unnoticed, leading to tragic incidents. To address this challenge, the World Health Organization has proposed a reliable, new HPTLC-based method for rapid screening of DEG and EG in cough syrups. So, why HPTLC for DEG and EG detection? Because firstly, it is recommended by WHO. You can analyze more than 10 plus samples along with the standard and QCs on a single plate within 90 minutes. It has a very simple sample prep. You weigh and you dilute, your samples are ready for analysis. It is cost effective and an orthogonal method to your GC. Importantly, CAMAG HPTLC can detect up to 0.05% of DEG and EG, which is below the regulatory limit of 0.1%. Proceeding further, I'll quickly show you in short how this analysis is done. As I mentioned earlier, the sample prep is very easy. You simply weigh 1 gram of a sample in 10 ml of a suitable diluent, methanol, and that's it. That's a solution ready. Now, we go and give command for our application by the help of this software, our VisionCatch software, where n number of data is successfully stored safely. We set the track assignment table before we give command, and here our samples and standards are placed in the vial rack. This is our HPTLC 60F254 glass plate, which we will be using for our analysis. We keep the plate on the plate holder and ensure to lock it. We close the lid and then go and give command via software. Once the application is done, now we move to the development. For development, we will be using the mobile phase, acetone, 25% ammonia, toluene and water in the ratio 85, 1, 5, 9.5, volume by volume by volume by volume. We will be using a Wattman filter paper number 1 for chamber saturation. Once the mobile phase is ready, we require 20 ml of mobile phase for a 20 by 10 HPTLC plate. We slightly tilt the twin trough chamber and pour our mobile phase. And after pouring a mobile phase, we tilt the twin trough chamber to an angle of more than 45 degrees. We do this just to ensure that an equal amount of mobile phase is being used for saturation and for development. And here, after a period of few minutes, our plate is developed. After development, we keep the plate for drying and go for dervatization. We use a suitable nozzle for dervatization. We will be using KMNO4 reagent to dervatize a plate. Once we insert a plate carefully into this dervatizing hood, we can then pour our dervatizing reagent. And then select the nozzle spray level and give command. This is exactly how the dervatization takes place inside the chamber. Once the dervatization is complete, take out the plate carefully. After taking out a plate from the derivatizing chamber, we will now heat a plate on CAMAG HPTLC plate heater. We will heat the plate at 100 degrees Celsius for 3 minutes. After heating a plate, we will now capture a plate image with the help of a CAMAG HPTLC visualizer. As you can see, a L-guard shaped tensile inside a visualizer. That's exactly where we will place a plate. And then go and give command from the software. And now this is the plate image captured after derivatization. As we look at the plate, you can see that track 2 to 5 contain the test samples. Track 6 and 7 are the spiked samples. Track 9 to 14 show the DEG and EG standards in decreasing concentrations varying from 20% down to 0.05% as per the WHO protocol. You will notice 3 well-resolved bands in the standards: glycerol at RF 0.37, the DEG and EG band at RF 0.51, and propylene glycol at RF 0.60. Now, if we focus on the test samples in track 2 to 5, there is no band at RF 0.51, which means they tested negative for the DEG and EG detection. However, in the spike samples from track 6 and 7, you can clearly see the DEG and EG band appearing at the same RF value, confirming proper recovery. And importantly, even the lowest standard at 0.05% concentration shows a visible band at RF 0.51. This concentration is below the regulatory limit, so the fact that we can detect it here demonstrates that the method is sensitive and capable of identifying even very low level of DEG and EG. So, why CAMAG HPTLC? To begin with, it fully supports this WHO recommended method for reliable DEG and EG testing. From a compliance point of view, the system is completely aligned with 21 CFR Part 11 compliant, which is an essential for pharmaceutical industry. Apart from 21 CFR Part 11 compliant, it is even compatible with clinical good laboratory practices and clinical good manufacturing practices. So every aspect of data handling and documentation follows strict regulatory standards. All data is stored securely with password protected encryption. The platform offers up to 7 user levels and 5 signature levels, giving you complete control over access, authorization and approvals. With these simple steps and rapid detection, we can reliably detect harmful components like DEG and EG in cough syrup samples while fully adhering to pharmaceutical regulatory requirements. CAMAG HPTLC ensures accuracy, reliance and confidence in every analysis. With that, we conclude this rapid detection of DEG and EG from cough syrup samples. For any further query, please contact us on lab@anchrom.in. Thank you.